So good evening, good afternoon, or wherever you are. It's my great pleasure to introduce today's speaker, Professor Patrick Ong. So he did his master's in applied mathematics and PhD in applied mechanics from the Delft University of Technology in 1993 and 1998, respectively. After postdoctoral stints with Professor J.W. Hutchinson at Harvard and Penn State, he joined the University of Groningen in 2003, and he is now a full professor at the Zurich Institute for Advanced Materials. His research involves computational physics, molecular biophysics, molecular dynamics, applied physics, and material science. So he's a member of National Committee Physics of Life. He is also a member of the General Assembly of IUTAM. He is editorial board member of International Journal of Molecular Sciences and also the Extreme Mechanics Letters. So these are the two journals. And so Dr. Professor Patrick Ong has published more than 170 research papers. And he is going to talk about the molecular dynamics of intrinsically disordered proteins. And for those of you who are new to this field, proteins are the molecular machines. And the conventional view is proteins have well-defined three-dimensional structures, and that helps the proteins to perform their function. But intrinsically disordered proteins, these are a very fascinating class of proteins, and they don't have well-defined structures, and they are involved in many important biological processes. I think I will stop here. And so title of today's talk is MD of Intrinsically Disordered Proteins. So Professor Ong, so over to you.
Okay.
So I'm sorry. So just for the participants, if you have any questions, you can please leave in the Q&A box. So towards the end, we can have interactions after the talk is over. Thank you. So Professor Ong.
Okay, thank you very much, Sankar. Thanks for this very kind introduction. I think Corona doesn't have many advantages, but there are only disadvantages. But one advantage is at least this webinar series. So it's a very nice opportunity to share this with people all over the world. So you are welcome now in my home office, right? So basically, where are we? So we are in Europe, right? More specifically in the Netherlands. And if you zoom into the Netherlands, then we are way up in the north, right? It's about 2 hours from Amsterdam, where you come in with your flight. So Groningen is established in 1040, where the first, evidence of the name Villa Groninga was appearing in a letter. It has 200,000 inhabitants. So for most of you, this is not a very big city, but for Dutch standards, it's still a medium-sized city. It has 60,000 students, of which 35,000 are living in the city itself. So it's clearly a very lively student city. Well, Groningen as such is typically Dutch, right? With the canals, with moderate summers, so highest highest degrees in the 20-25. Also have moderate winters, right? Where every year we have some snow and some ice. And that also allows us to do our favorite hobby, right? Which is ice skating on the canals, right? So the University of Groningen is established in 1614. It's part of the top 100 of best universities in the world. It has 33,000 students, and it is the home of 4 Nobel Prizes. And today's topic, as Sankar already nicely introduced, is on the nuclear pore complex, and which involves intrinsically disordered proteins. And here you see an image of this it's a big molecular complex sitting inside the membrane of the nucleus of our cells, and it's responsible for the transport of all molecules going in and out. Well, you might now think that, well, I'm misplaced, right? I'm in the wrong session, because the question is, this is a mechanics of materials webinar. So what does this have to do with mechanics of materials? I mean, there's no mechanics. There's clearly not a material. So, well, what I will show you, and I hope to show you at the end, that there's a resemblance, there's more resemblance than you might think at first glance. So to set the stage a bit, let's look at a typical example from micromechanics of materials, which is on cellular materials. So then you can identify 3 different length scales, right? And at the micro scale, you can identify individual struts. Then at the meso scale, you can identify how these single struts are organized in a 3-dimensional architecture, the cellular architecture. And then at the macroscopic scale, that's the scale of the applications. And their macroscopic overall properties are important, like stiffness, strength, ductility, toughness, etc. Right? And what we are typically aiming for is to find the relations, or structure property relations, that can relate the overall properties to the underlying microstructure at the microscale and at the mesoscale. And as a tool, we typically use multiscale modeling, right, to try to bridge the length scales all the way from the single strut level to the level of the applications. Well, I mean, I would like you to keep this example in mind when we go through the talk. And in the end, I will come back to this specific example and see if we can see some similarities there. Okay, so now let's look at proteins. As Chairman Shankar already said, I mean, proteins are basically the workhorse molecules of life. So they're involved in almost every structure and function in the human body. And proteins are basically long chains, polypeptide chains of 20 different amino acids. And depending on that specific amino acid sequence, I mean, there are 20 different amino acids. So depending on that sequence, it can form secondary structures like beta sheets or alpha helices. And then at a larger level of organization, they can form 3-dimensional tertiary and quaternary structures. And it has long been thought that proteins need to fold in a specific structure in order to do their function. However, nowadays, more and more evidence has appeared in which also intrinsically disordered proteins play an important role. And here you see an example of a protein that has partly a folded structure, a really ordered structure, and partly regions that are intrinsically disordered. And it now turns out that also this intrinsic disorder can play a very important role in all kinds of applications, right? And today I will talk about one of these applications, and that is in transport through the nuclear pore complex. And here you see the disordered proteins in green, and they basically line the central channel of this big protein complex. So if you look at a cell on a substrate and you would zoom in towards the nucleus, so you go through the external membrane, through the cytoplasm, and then you're approaching the nucleus. And then you see these big protein complexes in blue, and these are the nuclear pore complexes. And they have a typical 8-fold symmetry. And if you zoom in further, and then you can make a cross section, and then you can clearly see that it consists of intrinsically disordered proteins, right? And these are called FG-Nups. And they're called FG-Nups because the protein family is called nuclear porins and abbreviated by Nups. And they're called FG-Nups because they are rich in two kinds of amino acids, namely phenylalanine and glycine, right? And it turned out that the only way that molecules can actually enter and leave the nucleus is through the nuclear pore complex. And small molecules, they can basically freely go in and out, but bigger molecules, they are excluded. So bigger molecules larger than approximately 20 to 40 kilodalton, so that's a certain mass, they are excluded. And the only way that big molecules can actually enter is when they bind to a nuclear transport receptor, NTR in short. Right? And these NTRs then take that molecule and bring it through the nuclear pore complex. Well, we know that this works, right? But what we don't know is how that specific transport mechanism is mediated by these FG-Nups, right? And this is what we are aiming for in this work. And to try to shed light on this, we developed a multi-scale modeling approach. And the aim of that approach is to bridge the scale from the amino acid sequence to the overall conformation of the nuclear pore complex. And to do that, we built a model which is basically consisting of a polymer chain based on beads that each bead represents an amino acid. And the model is fine enough, right, so that it can account for the exact amino acid sequence and the properties of the individual FG-Nups. But on the other hand, it's also coarse enough so that it can account for the collective behavior of all these different FG-Nups that line the central channel of the nuclear pore complex. So if you, the one bead per amino acid model, as we have called it, is basically a model where the all atomistic polypeptide chain that is depicted here is mapped on a coarse-grained chain, right, with the coarse-grained beads located at the C alpha atoms, right? So then by making this scale transition, so making this coarse-graining step, we go from a very detailed energy landscape, right, to a much more coarse energy landscape, which is much smoother, right? And by doing so, I mean, you don't have to account for all these details that you see at the atomistic level. And of course, you reduce enormously the amount of degrees of freedom, right? And therefore you can look at much bigger systems. So the model accounts for bonded potentials that account for stretching, bending, and torsion, right? So that is the bending, stretching, and torsion of the backbone. And it accounts for non-bonded interactions to account for electrostatic and hydrophobic interaction. And the water is implicitly accounted for through Langevin dynamics. So here you see the hydrophobic potentials. So this is the description. And to account for these hydrophobic interactions, we have an interaction parameter, epsilon ij, where this is the interaction parameter that sets the strength between an amino acid I and an amino acid J. And the input for this are the normalized relative strengths of each individual amino acid. And that is taken from the experimental hydrophobicity scales. So here we have 20 different values that are experimentally introduced, right? And here, and by doing that, we can basically go from a very attractive potential, and that is the case where you have two purely hydrophobic amino acids, and it can go to a more repulsive state. So this is very attractive, and this is more repulsive when you combine two hydrophilic amino acids, for instance. And in addition to that, we have electrostatic interactions, which we account for through a modified Coulomb law that account for solvent polarity and ionic screening, right? So these kind of interactions are explicitly taken into account. So then we went doing simulations, and then we compared the results to experimental measures on the hydrodynamic radius, or the Stokes radius, developed in the group of Rexach. And here we compare the experimental hydrodynamic radius with the predicted one by the model. I mean, we used two NupX to set the free parameters in the model. We still have a couple of free parameters. And then we predict the Stokes radius for the rest. And that gave us an average error of 7.5% and a maximum error of 17.5%. And that is, given the uncertainties in the model, is an accuracy that we were satisfied with. So then in the next step, we used experimental data to build the scaffold. So we built a coarse-grained computational model for the scaffold. Then we used experimental input on the anchor sites of all the individual FG-Nups, and we used that to anchor the individual FG-Nups in the scaffold. And all these different FG-Nups, so we account for the 10 different FG-Nups inside the yeast nuclear pore complex. And as you can see here, every NUP has a different length and a different amino acid sequence, and every specific FG-Nup is anchored at a very specific anchor site, right? So we take all this complexity into account, and then we basically run the simulations, right? And here you see a typical example where two FG-Nups are highlighted, and that is to emphasize the different length, the different location. So you see the green ones is much more comprised in the center, so it's much more confined, and the red one is much more extended, and we know that is because they also have a different functional role in doing so. So then if you take snapshots of these amino acids in time, then you can look at a density distribution. So we looked at the time average density distribution. And then if you look at all the amino acids, then you see that you can guide of a donut shape with a low density in the center. If you only zoom in at the charged amino acids, then you see that these are basically localized at the periphery of the channel. And if you then zoom in on only the FG repeats, so right, those are the specific repeats that are essential for nuclear transport, then you see that they also form a high density region more in the center, right? Well, the nice thing about doing computations is that you can also play a bit with the amino acid sequence. So what we have done is to quantify, we looked at the circumferential averaging, right? So there, with that, you can create a radial density plot, right? So here you have the radius, and here you have the z direction, where we have averaged in the theta direction. And here you see this typical footprint of this high density region, right? Forming this donut kind of shape. And then we looked at a case where we basically replaced all the charged amino acids with neutral amino acids, right? And then what you see is that all the FG-Nups basically collapse inside the pore. So they form a big high density cluster near the sides of the scaffold. Then if you then in the end also mutate away also the hydrophobic amino acids, and you also replace them also with neutral beads, then basically you have a denatured nuclear pore complex where the distribution is very uniform in the pore, and basically all the FG-Nups are expelled from the core of the pore. What we can also do is we can look at specifically at the role of the amino acid sequence. So here what we have done in this case, we basically have taken all the FG-Nups and we reversed their anchor site. So instead of anchoring them at the C terminus of the amino acid sequence, we anchor them at the N terminus, right? And then what you see is that you lose that specific footprint of this high density donut-like region. Here we looked at the case in addition where we simply put all the amino acids, we shuffle them around, and then we uniformly distributed the hydrophobic amino acids and the charged amino acids along the length. And in that case, all the amino acids are there, as in the reversed case, But then also you see that the specific high density regions, they vanish, right? So this clearly shows that there is information hidden in the amino acid sequence that determines the presence of this high density ring. Okay, then we did some comparisons with experiments. So here you see the results from single molecule experiments in living cells carried out by the group of Weidum. And then what you see is that if you look at inert small molecules, then these small molecules that are colored in red, they basically go through the central channel. But the nuclear transport receptors, so these karyophrins, they basically go to a different region, and these different region in green basically co-localizes with the high density region where we see a lot of FG repeats, right? So that makes us, so combining the density distribution and the single molecule experiments, that basically made us conclude that there can be passive transport, right, through the central region. And then there's facilitated transport through the high density FG ring by the interactions of the NTRs with the FG repeats, right? So that is what we can conclude from the density plots alone. So then we looked at a set of data from the lab of Susan Wente. And what she did is she looked at mutations. So they looked at yeast cells, and they mutated the yeast cells in such a way that you can remove a considerable amount of FG-Nups. So then she observed that half of the FG-Nups can be removed, and then the cells are still viable. On the other hand, if you use specific combinations of FG-Nups and you remove those, then it turns out that these cells are no longer viable. So these cells die, right? So what we wanted to do is to, okay, let's see what happens if you mutate away those specific FG-Nups and see how that is affecting the structure of these amino acids inside the NPC. So that is what we have done. So here you see the case, here you see one viable case. And this is a viable case that we have chosen because here a lot of mass is reduced, right? So almost half of the mass is reduced, but you still see the specific footprint of these high density regions, right? Which is really resembling the wild type FG-Nup, the wild type NPC. And here you see 5 cases of inviable yeast strains. And here in these cases, you see that the high density regions, these have vanished, right? So to quantify this, we looked at the radial density distribution. So here you see on the vertical axis that the density as a function of the radius, right? Where radius is 0 is the central axis, right? And then what you see is that the viable cases, right? They have a density region with densities larger than 180 milligrams per milliliter. But the inviable cases, in those cases, all the densities remain lower than these 180 milligrams per milliliter. So we cannot say more about the complexity of all the transport, but the only thing that we can say is that the viability is correlated to the presence of this high density ring, right? So you need a density larger than this 180 milligrams per milliliter in such a way that you have viable yeast cells. So that says something probably on the facilitated transport, right? Where we have these high density regions. But now let's zoom in on the passive transport. And this is work done in collaboration with the lab of Lisbeth Veenhoff in Groningen at the University Medical Center. And she is studying transport in yeast cells. And she took a couple of the strains from the lab of Susan Wente, and she used strains that were viable, right? Because otherwise she could not find a readout. And then she built different constructs of different sizes, and then she looked at the transport rates of these constructs. And what we done is we have simply taken those specific yeast strains and we deleted the FG-Nups that were also deleted in the mutated cells, and we calculated the density distributions. And then to summarize again, we look at the radial density distribution. So here you see these typical footprints. And what we see is that for all these strains, the densities at a certain region is larger than this critical 180 milligrams per milliliter. So that confirms the fact that these cells are indeed viable according to our correlation with density. And then having the single molecule experiments of Weidong's group in mind, where passive transport goes to the central channel. So then we looked at the region in the central channel. So we identified a cylinder with a diameter of 10 nanometers. And then we asked the question, I mean, can we correlate the permeability that is measured in the yeast cells of Lisbeth Veenhoff with the density that we calculate in the central channel of the nuclear pore complex? Well, and the answer is yes. So here we plot the average density in the center from the computations, and we set it out against the transport rates that are calculated from a node that are measured in the yeast cells in vivo, right? So we can actually compare in silico results to in vivo results. And here you see a clear correlation, right? So that makes us conclude that the in silico density correlates with the permeability barrier measured in living yeast cells. Okay, so this work was nice, and it really showed that on the one hand, that, I mean, the yeast nuclear pore complex is highly complex, right? So there are 10 different FG-Nups, different lengths, different amino acid sequences. But what we do find is that we can correlate the structure to experiments in terms of passive transport and active transport. However, what we really wanted to do is really going more on the physical mechanisms, right? So based on the observations that there is a balance between charged amino acids and hydrophobic amino acids, and that balance between charge and hydrophobicity, that basically dictates the structure of the proteins inside the NPC. We have seen that the amino acid sequence here plays an important role. We have seen that these protein distributions correlate with passive and facilitated transport, but we have also seen that many of these FG-Nups are redundant, right? So you can lose half of the mass of the NPC, where still the cells are viable. So still you will have transport in and out of the nucleus. So that made us ask the question, so then probably, so there probably is a minimal NPC. So if you lose a lot of complexity, so what is the essential mechanism that actually dictates the transport? So that makes us move into a different direction, and following instead of a top-down approach, we follow the bottom-up approach. And here we collaborate with the experimental biophysics group of Kees Dekker at the Delft University of Technology. And here in the lab, they were able to make biomimetic nanopores. So they create solid state silicon nitride films of 20 nanometer thickness, right? And then they functionalize these nanopores using a native yeast FG-Nup, right? And this yeast FG-Nup is NSP1. And to investigate the role of hydrophobic amino acids, we also looked at a strain where all the hydrophobic amino acids have been mutated away and replaced with serine, which is a hydrophilic amino acid, right? So there we have one native FG-Nup and one mutant where the hydrophobic amino acids are removed. And then they make they apply a bias voltage, and they establish an ion current over the channel. And then they put in two different kinds of particles. So they put in T-cherry, which is basically an inert protein of a certain size. And then they put in a nuclear transport receptor, this Cap95. So that Cap95 is one family of the caryoverins nuclear transport receptors, right? And then by looking at the traces, so here you look at the ionic current traces, then every time you see a downward spike, then you know that one of these molecules are transporting through the pore, right? And then, then you can use that event frequency, right? So here you see the event frequency for different pores, and then you can see whether the pore is selective or not. And it turns out that if you look at the event frequencies for the native pore, so that has only one NSP1 FG-Nup, right? Then the Cap95, so the nuclear transport receptor, that is actually going through, right? So you have a high event frequency, as high as for the bare pore, but T-cherry basically is excluded, right? So that means that here you have a selective pore, right? So particles that do not have a role inside the nucleus, they are basically excluded, and only the nuclear transport receptors that are important for carrying cargo towards the nucleus or out of the nucleus, they are able to go through. On the other hand, if you mutate all the hydrophobic residues out, then you don't see any difference in transport rate between the nuclear transport receptor or the inert molecule. So that means that the molecule that should not be in the nucleus is also transported. So that means that this pore is not selective, right? So to shed a bit more light on that, we also performed simulations. And then, so we carried out umbrella sampling for the particles along the central axis. And that gives you the potential of mean force, right? The potential of mean force along the central channel. So here you see the potential of mean force along this Z coordinate. And that gives you an idea of the energy landscape that the T-cherry and the Cap95 see when they travel through. And that is the energy landscape that they have to overcome for translocation. So what we see is that, that for the T-cherry through the NSP1 pore, there's a very high barrier. There's a very high energy barrier for transport. And if you compare that to the barrier for Cap95, so the nuclear transport receptor, here you see clearly a big difference, right? So from an energetic point of view, the NSP1 pore is selective. On the other hand, if you look at the energy profiles, the energy barrier for Cap95 and T-cherry in the mutated pore, then you basically see that they have a similar barrier. So it cannot distinguish between transport or non-transport. So to make a comparison to the experiments, so we used an Arrhenius relation. So here you see the Arrhenius relation to relate the event rate to the energy barrier, delta epsilon, right? And then when we make a comparison, so here you see the event frequency for the different pores, and you compare experiment to modeling, then you clearly see that you have a very good agreement. So both in terms of experiments and in terms of modeling, we are able to show that where you have a native FG-Nup, so you only one native FG-Nup is sufficient to actually create from the bottom up a functional nanopore, right? And that modeling, both modeling and experiments, support the selective nature of this pore. So then we went one step further, right? And then we looked at bottom-up design of NPC mimics comprising designer FG-Nups. And then we basically asked the question, I mean, instead of having one native FG-Nup, can we create an artificial FG-Nup with an amino acid sequence that is created from scratch and still create a selective nuclear pore complex. And if we can do that, then we have full control over the amino acid sequence, and then we can start playing with the amino acid sequence and see which kind of properties in that sequence plays an important role in mediating the transport. So that can give us information on the essential and the minimal properties that are needed for selective nanopores. So what we've done is we took a number of essential FG-Nups, and that are depicted here. And these essential FG-Nups, they have an extended domain in red and a collapsed domain in green, right? And we look at the statistics of these different domains of these essential FG-Nups. And based on that information, we created an artificial FG-Nups, right, which we termed NupX, that consists of a collapsed domain and an extended domain. And here you see the explicit amino acid sequence of NupX. And in both the collapsed region and the extended region, we used a charge to hydrophobicity ratio and a spacing between the FG repeats that is equal to the average of these essential FG-Nups. Right? So then we carried out experiments. So in a similar setup as what I have just shown, using ion currents to look at the event rates of transport. And parallel to that, we also carried out coarse-grained molecular dynamics simulations with our model. And here you see an image simulation of this, where we have the nanopore. We used surface plasma resonance to find the spacing between the anchor sites of the FG-Nups. We placed the FG-Nups, and then we carried out simulations for different diameters. And then you see that the morphology of the NupX-coated pores is highly dependent on the pore diameter, right? So with pore diameters that are smaller than 20 nanometers, then the FG-Nups are basically expelled. On the other hand, if the diameter is larger than 45 nanometers, then the density is much smaller and you create a hole in the center. And therefore we have chosen an intermediate size where we are still having a closed pore, but we're intermediate to the regimes where the FG-Nups are expelled and those where you have an empty region in the center. So for both the experiments and the modeling, we used a diameter of 30 nanometers. So then we analyzed the transport of Cap95 and T-cherry. And for Cap95, we created basically a sphere, right, which has an inert surface, but we do account for the averaged amount of charge on the nuclear transport receptor, and we do account for a strip of hydrophobic binding sites that have also been observed in all atomistic simulations of that specific Cap95 structure. And then when you put this in, then you see that these nuclear transport receptors, these Cap95s, they can actually enter the meshwork, right? And here you see 3 different snapshots at 3 different times. So initially the caps are placed in the box above, and then during time, they start interacting with the FG-Nups and they're able to go through. On the other hand, when you only have inert cargo, so no charge and no hydrophobic binding sites, then basically these cargo, right, so these T-cherry particles are not able to enter, right? And that is due to the fact of the steric hindrance, right? So since they do not have any specific interactions with these FG-Nups, they're basically excluded because of excluded volume interactions, right? So they are not able to enter. So conclusions on the work of NupX. So both based on the experiments and the modeling, we conclude that NupX faithfully mimics the selective barrier of the nuclear pore complex, right? So it can establish a permeability barrier so that particles that are inert are not able to enter, but nuclear transport receptors that feature a charged surface and hydrophobic binding sites, they are explicitly able to enter. Well, this approach opens a platform to examine more in detail the relation between the FG-Nup amino acid sequence and NPC selectivity, right? And that is what we're currently already working on. So we're currently working on NupX 2.0, and there we focus on the effect of the ratio of charge over hydrophobicity, and we focus on the FG spacing. So here you see already some initial results. So here you see simulations that we have carried out for different combinations of FG spacing and charge over hydrophobicity ratio. And by varying the spacing between the FG repeats, you can control the density of the sites for NUP NTR interactions, right? So to facilitate active transport of the nuclear transport receptors. And perpendicular to that, we have varied the charge over hydrophobicity ratio, and that controls the density, right, of the NUP meshwork. And that density we know is essential to establish a permeability barrier so that inert cargo is not able to enter. So that brings me to an overall conclusions, right? We looked at 3 different levels of analysis. We started out with the yeast NPC, where we have 10 different FG-Nups with different amino acid sequences anchored at different locations in the complex scaffold of the NPC. We have seen that the structure of the FG-Nup distribution is in a donut shape, and it's encoded in the amino acid sequence, and it's determined by the distribution of charge and hydrophobic amino acids. The permeability barrier is correlated to the density in the central region, right there in a region where the radius is smaller than 5 nanometers. We found that the viability of yeast cells is correlated to the presence of a high density ring, and then the critical density we found was larger than 180 milligrams per milliliter. Then we made the step from top-down analysis to a bottom-up analysis, looking at biomimetic nanopores that only comprise 1 native FG-Nup instead of 10. And based on the experiments and the modeling, that the selectivity of the nuclear pore complex can be mimicked in vitro, so in the lab, using this artificial nanopore. And we found that if you mutate away the hydrophobic residues, then that destroys the selectivity. So the hydrophobic residues are essential for creating a selective nanopore. And finally, we went 1 step further. We looked at nanopores comprising only 1 FG-Nup, but then we de novo designed the FG-Nup sequence, right? So we have basically mutated E. coli bacteria, right, to generate these kinds of FG-Nups. And we found both based on experiments and modeling that with an average native composition, right, comprising a collapsed domain and an extended domain, we can also capture NPC selectivity. And now this is successful. This opens the platform to explore more fundamental mechanisms that drive transport. So for instance, is the spacing between the FG repeats, right? Does that correlate with the spacing of the FG repeats on the nuclear transport receptor? So that is what we're currently modeling. So we're now modeling a more refined representation of the CAP95, where we have an atomistic representation of the amino acid sequence. And then we discretize that using our coarse-grained model, where we have 1 bead per amino acid. Okay, so that basically concludes my talk, but I promised to come back to the initial question on what this has to do with micromechanics or mechanics of materials. So it's clear that from the talk, from cellular materials at the microscale, we have single struts, but if you now replace that with a single FG-Nup, then at the mesoscopic scale, we had the cellular architecture, right, with information how these individual NUPs are basically connected together to fill a three-dimensional structure. Well, that is basically replaced by NPC structure. And at the macroscopic scale, we looked at structural component where important properties were stiffness, strength, ductility, and toughness. But now the car can be replaced by the cell nucleus, right? Where nuclear transport is mediated by hundreds of these nuclear pore complexes, right? And instead of stiffness, strength, and ductility, we basically have permeability barrier, facilitated transport, and measures for selectivity. So the aims are the same, and basically the tools are the same. So in the end, these two problems are not so different after all. So finally, I would like to acknowledge my current group members and also my former group members and my collaborators. And specifically for the talk of today, I would like to especially thank Ankur Mishra, Henry de Vries, Ali Kavami, who basically initiated the one-bead-per-amino-acid model, and my collaborators, Eric van der Giesen, longtime collaborator and former promoter, Lisbeth Veenhoff, collaborator at the UMCG, and Kees Dekker at Delft University of Technology. And I would like to thank you for your attention.
Thank you, Professor Monk. So I see that there is one of the participant has raised his hand. So maybe we'll take up the question one by one. Is it fine with you?
Sure.
Yeah. So Mr. Manjesh Singh, so can you, yeah, please go ahead. Manjesh? You are not audible. I don't know, maybe you have to unmute. Or maybe you can type your question. Okay, he says, I don't see unmute option. Then please type your question, then I will, yes, can you unmute me? So I don't, I have to request.
Yeah, Shankar,
yeah.
You can unmute him.
I can, just 1 second.
We just go beside his name and...
Yeah, I'm doing that. I will. But I don't see his name. Yeah. Yeah, please, yeah.
Yeah. So, am I audible now?
Yes, you are audible now. Yes.
Thanks, Professor Ong, for a nice talk. I have a few questions. Pardon my limited understanding of biophysics. So, first of all, this one bead per an amino acid. So, how do you take care of this side group? The bulkiness of the side group in terms of persistent length and all in this coarse graining.
Yeah, no, in the results that I've just shown, we do not take that into account. So, the geometrical presence of the side chain, so of the residues, is not taken into account. So all that information, including its hydrophobicity and its charge, is all concentrated in one bead located at the C alpha atom along the backbone. But in a model that we are currently working on, there we do take that into account. So we're currently working on an extension of 1 BPA to 4 BPA, right? Where now in addition to the beads at the C alpha atoms, we also have a bead that represents the explicit residue. And by playing with the distance from the C alpha and the radius of the bead, then we can account for the different size of the different residues. And on top of that, perpendicular to this backbone, we also have additional beads that can account for hydrogen bonding, right? And hydrogen bonding is important when you make the transition from intrinsically disordered to ordered structure, right? Because then beta sheets, right, one of these secondary structures, these are very important, and that is what we're currently looking at self-replicating molecules, right? And we are looking at liquid-liquid phase separation and also amyloid formation, right? So that is something that we're currently working on. So your question is very relevant in that respect.
And can I ask a couple of more questions? Actually, I have, it's quite interesting to me. So I have a couple of more questions. Can I ask or?
Yeah, please go ahead. Yeah.
Okay, so and next question to just take it forward about this intrinsically disordered protein. So I mean, normally when we model something like coarse graining or all atoms, so we have some structure factor which we compare. So or experimentally also we can get those structure factors or form factors. But here in case of intrinsically disordered protein, so I saw that you are comparing this hydrodynamic radius, but that can be because we are doing this structure property relationship, the same hydrodynamic radius can be for different kind of structures.
Yes.
How do we validate this model exactly? I mean, in terms of when compared to experiments.
Yeah, so that goes on two levels. So first, the input for the model is based on experimentally calibrated hydrophobicity values for the individual beads, right? So that basically allows us to change, so to capture the different hydrophobicities of the 20 different amino acids. That still leaves a couple of free parameters. And then we used these experiments on the hydrophobicity, so of these hydrodynamic radius, to pinpoint, so to fix those remaining 3 parameters in the model. And then we predicted the Stokes radius or hydrodynamic radius for the rest. And then we get quite a good correspondence. So based on one hand, the 20 different experimental hydrophobicity values and the calibration with the Stokes radius at a larger length scale of resolution, that gives us confidence that we are picking up the correct physics.
So I mean, okay. And that last one, if I can ask one more about that free energy barrier curve, which you had as comparison of 4 different kind of, I think, sequencing. If you can go to that slide. So the one where there's this.
So you are talking about the potential of mean force profile, this one.
No, not this one. The free energy or the energy barrier. Yeah, this one. So when I think of energy barrier, am I wrong to think in the sense from the valley to the peak?
Yes.
So I mean, in that case, is there? Okay, no. I mean, so I misunderstood then. So I mean, I was thinking in terms of that. Can we say that this black one has higher energy barrier in comparison to the red one or the green one? If you compare the peak and the valley.
Yeah, so what we have done is we basically looked at, so you go from the 0 line, and then you basically looked at the difference between the minimum and the maximum. So if you do that, then here you get a barrier of 6.4 and 6.5. And if you do that for this profile, you get a barrier of 6 kilojoule per mole, right? So it is comparable, but it's slightly smaller.
sorry, I didn't see the numbers. But thanks a lot. It was really nice. I'm done. For someone else.
So, Manjesh, did you? Okay. So I think Sumit also has a question. So, Professor Sumit Basu.
Patrick, thanks
Yes.
for the nice talk. I have a question about a figure where you compared the permeabilities with experiments. Yeah, so in this coarse grain, yeah, this one, yeah. So in the coarse grain simulations, time in the coarse grain simulations would be different from time in reality, right?
Correct.
So how do you compare permeabilities?
Yeah,
Because
so-
they're rate quantities, unless you do a scaling of time, how do you compare permeabilities?
Yeah, so you're absolutely right. So basically, the physical dimensions of time are coarse-grained out, right? So the only thing that you can do is you can look at time averaged conformational quantities, right? And that is, in this case, what we have done. So the only thing that we are comparing here is protein densities, right? So the only thing that we are comparing on the horizontal axis is the average density in the center from the simulations, and we compare that to the corresponding experimental measured transport rates.
Okay. Yeah, Shankar, I am done.
I think Shankar is frozen.
Hi, Shankar. Oh, he's.
Yeah, I think he is.
Oh, he has.
Can you take over moderating? Yeah.
I can't take over unless he allows me to. Oh, I am the host now. Okay.
Yeah, probably he will come back in.
Yeah, he said he had some questions. So in the meantime, are there? Okay, he has come back. Ratna is there in the audience, so he says hi.
Hi, I'm sorry, I lost my connection.
Yeah.
I don't know how. Yeah, so I have a question about your potential of your potential energy function, where you have a term for hydrophobic interaction, which is a modified Lennard-Jones potential. And this hydrophobic interaction depends upon, it is basically, it takes values from the hydrophobicity scale. And there are like 100 different hydrophobicity scale. And which one you have used, and how sensitive it is for the choice of hydrophobicity scale.
Yeah. No, you're absolutely right. I mean, there are many different scales. So what we have done is we looked at many different scales and we basically averaged them. So we normalized them per scale. We normalized them and we compared them, and then we renormalized again, which we think is like an average results, but these are still susceptible, I mean, to differences, right? And what we are currently also doing is we are comparing also with O-atomistic simulations. So then we basically do umbrella sampling for the interactions using O-atomistics for individual amino acids. And And then we can see how that compares to the values that we use here.
Okay, thank you. And so how many naturally occurring, NupX, how many of them are there? I think you showed in yeast there are 10. Is it, am I correct? Or like, for example, in humans, how many are there?
Yeah, no, there are more.
Okay.
So I think in yeast there are 12 or 13, and I think in humans it's approximately the same.
Okay.
Because you can identify homologs, which have
Mhm.
approximately the same amino acid sequence, but we
Okay.
took only the ones for which we knew the location of the anchor sites.
Okay. And I mean, in your model, you have the lipid bilayer, which is the coarse in the coarse-grained model, and the FG-Nups are simply placed inside. With the, like, how do you determine the density? And second thing is, since these are intrinsically disordered proteins, they come directly in contact with the lipid hydrophobic part of the lipid bilayer. So, which is probably energetically not favorable situation, because their backbone functional groups will come in contact with the acyl chain. So, how do you take care of these factors?
Yeah, very good question. So first, for the yeast MPC, we actually know the locations of the anchor sites, because at the end of the FG-Nups, there's also a folded domain. And the folded domain that they can see using their experimental techniques. So we knew in the yeast MPC, within, of course, certain accuracy, we knew the locations. Of course, for the nanopores, so the biomimetic nanopores, we don't have that information, but then we used surface plasmon resonance experiments. There you can find the areal mass density of the NupX, and from that you can back out what is the average spacing between the FG-Nups, and then we simply used a fully triangulated distribution of the anchor sites with an equal distance between every FG-Nup, and we put them there.
Okay.
So that is
I have a few more questions, but I will ask only one more question. See, in your model, you have this the lipid bilayer. Are you constraining the lipid bilayer, and only you are inserting this FG-Nups inside this the ring, or do you also allow the lipid bilayers to move so that you are going to have variable size? And so how do you do in your simulations?
Yeah. No, again, this is a very good point, because it is known that even viruses can pass through the nuclear pore complex, and viruses have a diameter that is larger than the central region of the nuclear pore complex. So the nuclear pore complex is compliant, so it is able to accommodate its size, and of course that is due to the fact that it's embedded in the lipid bilayer. Did not account for any of those effects. So in our case, it is a rigid cylinder to which we. So,
but why sorry, Patrick. Why did you then have so many atoms in the lipid bilayer if it's a rigid wall?
We don't model the lipid. We don't model the lipid bilayer. The only thing that we model is the nuclear porins, right? Which form the scaffold inside the lipid bilayer. Right, so only the proteins, which are folded proteins that are anchored in the lipid bilayer, that is what we use as an anchor site on which we put the FG-Nups. And of course, that also screens the interaction with the hydrophobic core of the lipid bilayer, right?
Yeah.
So therefore, we don't.
Maybe I will ask one last question. Although you have mentioned the role of, the designed FG-Nup, where you talk about the role of charged and hydrophobic. Where what you did not mention is whether this particular system is selective for, like for example, positively charged or negatively charged, or simply it is any charge is acceptable. Because I think in another case, when you mutated to all serine residues, then it lost its selectivity. So when you say charged, naturally occurring FG-Nups, so do they allow only substances which have very specific charges, or there is no charge specificity?
No, we did find in the yeast NPC, we did find that charge is also important. So we looked at, for instance, NTRs, caps, that do not have hydrophobic binding sites, but only have charge. And only with charge, these particles are entered to go in, right? And that is also consistent that when you look, if you compare the NTRs with average proteins, then it turns out that these NTRs have a much larger charge to hydrophobicity ratio, which suggests that charge plays a role. In addition, the NTRs have a net negative charge, while the FG-Nups have a positive charge. Therefore, we also
complementary.
There's a role, right? There's a role also for charge interactions there.
Okay. Thank you. It's a very, very exciting talk, and it's very much relevant to what I have been working, and I mean, all of us, we really enjoyed your talk. I don't know whether there are any more questions among the participants.
I just wanted to ask Patrick, these simulations from one of your slides, I thought I saw that these are microsecond long.
Yes.
Oh, that's very long for an MD simulation, right? I mean,
yeah. I don't remember where this is present.
I think towards the end, where you were showing the cap 95 particles.
Yeah, so these are probably microseconds, right? Yeah, microseconds. Yeah. So we typically do a couple of microseconds, but again, we did not make a clear one-to-one calibration run to relate this time with real time. So it should be taken with a grain of salt, but I think it will be around this order of magnitude. But we basically use it for ourselves to really look whether the results are converged, right? So we have to take, I mean, we need to sample sufficient so that we can have statistically averaged properties.
And how long does a 2 microsecond run take on the wall clock?
Yeah, so that will typically take, well, maybe a system like this, a couple of days or to a week, maybe 2 weeks.
Okay.
Yeah.
So just one last.
There's still big calculations. Even though it's coarse-grained, there's still, I mean, very big calculations.
Yeah. 2 microseconds is very long.
Many different cores.
Just one last question, curious. Have you used this hydrophobic potential to study protein folding?
So as I explained, I mean, we are currently looking in protein folding, not with the 1-BPA, but with the 4-BPA model. Then we have some initial success. So in the simulations that we have seen, we're also looking now at amyloid formation. So that is in neurodegenerative diseases. Then initially these proteins are disordered, but
and then
some mutations, they become ordered, and then they form these beta sheets and amyloid fibers, and then they cause all these protein plaques in the brains. And I think for the first time now we have a coarse-grained model that can look at very big systems, but is also to make the transition from disordered to ordered structures.
Okay. Thank you. So
Shankar, I think Manjesh has a last question. He has raised his
last
hand.
question.
Yeah.
Okay. So
I mean, there are a lot of interesting things, so I am not able to keep, I could not keep track of all the things, but now that we are back on these slides, so this sphere, I believe these spheres are rigid, right?
Yes.
Yeah, and they are interacting, so they're kind of, I mean, they're levitating. So, I mean, is it like this Langevin dynamics kind of implicit solvent kind of thing there that they're levitating, or?
Yeah, so gravity is not in. So these particles are so small and their density is so small that gravity doesn't play a role. So they're levitating because of the random kicks that they get from the Langevin simulations, right? So in Langevin simulations, there's this balance between random kicks that they get from the solvent molecules, right? That is also like a thermostat. So that is to account for a certain temperature. And then balancing that is viscosity, right? So that is the hydrodynamic interaction that is somehow accounted for. And then of course there are the enthalpic interactions, so the specific interactions of hydrophobicity and electrostatics.
And I
so
believe
if they are levitating, then they're basically floating in solution, right?
And I believe all these simulations are much above the glass transition temperature for these things, or?
Glass transition temperature of what?
Of this protein, like these chains, polymer chains of proteins.
No, I mean, these are biological proteins and they don't have...
They're in the solution, so I mean, there will be much lower this TG, I think.
Do they have a TG?
I mean, of course they will have, but because they're in solution, so it will be much, much lower than what you expect in general. Like even for polystyrene or something, it's around 100. But if you go to solvent, it will be down to, I mean, no, no, 10, 20 something. Depends on the concentration. So, and that was just a side question.
Okay, well, I mean, what we are currently also looking at is, I mean, there is a transition, right? So that the system remains disordered and it remains random. And then there's a transition to order, which also has a pathological consequence, right? Because if this is basically still a liquid, right? If this liquid makes a transition from a liquid state to a solid state, then in biology, that means that something goes wrong in the body, right? So in that sense, these kind of transitions are biologically extremely relevant. That is also, and what we think is that these nuclear transport receptors that are always there inside the pore, they keep it liquid. So because of all these interactions, they're called fuzzy interactions, they basically avoid that all these FG-Nups collapse together and form a transition to a more ordered solid.
But interesting.
It's a nice analogy as well. I mean,
just maybe one last question. So, I mean,
that's the last question. Yes.
I'm sorry. So, have you looked at the mean square displacement or dynamics of this one? So, are they like comparable to this a linear polymer chain kind of thing, or?
Well, I mean, the densities of these structures is much smaller, right, than polymer melts. But diffusion coefficient is a very important overall property, right? Because that also says something about the liquidity of the network. So we're definitely going to look into the diffusion coefficients when these proteins or these particles go through. And how does this diffusion coefficient depend on those specific interactions that these nuclear transport receptors make with these FG-Nups? And then we can also learn something about transport efficiency, right, based on the specific amino acid sequence. And those are really relevant quantities to look into.
Thanks. I look forward to meet you sometime and have some more discussion.
Yeah, no, same here. Same here.
Yeah. So thank you, Professor Ong, and I think I guess there are no more questions. So on behalf of the organizers, Professor Sumit Basu and his colleagues, and on behalf of the Mechanical Engineering Department of IIT Kanpur, I would like to thank you so much for this exciting, fascinating talk. We all enjoyed. Thanks for sharing your research. We really enjoyed and hope to see you here in the campus very soon. Now that the vaccine has been officially approved, hopefully it works. And so we look forward to meeting you here in the campus.
Yes, I also look forward to meeting you in person.
Right.
Okay.
Thank you, Sumit. Please
thanks a
over
lot,
to you.
Patrick. So it was a pleasure.
Thank you.
So,
Same here.
We'll call it a day. Close the meeting.